So welcome everyone. In this video we will talk about the theories of acid base indicators. Okay, so in the previous video we talked about theories of acid base titrations, and these are the theories of acid base indicators. Okay, so do not confuse between both of them. The theories of acid base titration are different and the theories of acid base indicators are different, and both of them are equally important. Okay, so now let us begin with this video. So first theory is the litmus theory. Okay, now what is happening here, that according to litmus theory, indicators are similar to litmus in their behavior. Okay, so you might have heard about litmus paper. Okay, so whenever we dip it in a solution, if it turns into blue color, we say that the solution is alkaline, and if it turns to red color, we say the solution is acidic. Okay, so the same litmus is being involved here. So what is happening, that litmus is actually, it is a weak acid which shows red color in unionized state and blue color in ionized state. Okay, so here we are talking about the ionization. So I will repeat again, that litmus, what happens, that in unionized state, which means that it is not broken down into ions, then it is having red color, but when it is broken down into ions, it shows blue color. Okay, so what happens, that the litmus solution in presence of hydrogen ions, which means that when we dip the litmus solution in acidic medium, there are hydrogen ions. Okay, so what happens, that it results in the formation of unionized litmus, which gives red color. Okay, so I hope this is clear to you. Whenever we talk about acids, what are acids? Acids are those which donate the H positive ions. Okay, so in an acidic solution there are more H positive ions, so that is why what is happening over here, that the litmus solution in the presence of hydrogen ion in excess amount results in the formation of unionized litmus. And just now I said that the unionized state is having red color, so the litmus turns red. Okay, so this is what happens when we put our litmus in our acidic solution. Whereas on the other hand, in presence of excess hydroxide ions, which means in case of bases, what happens, then the litmus turns into ionized litmus, it breaks down into ions, and the ionized litmus is having blue color. Okay, so this is the all the ionization of litmus which defines our color. Okay, so here we talked about the litmus theory, that in the unionized state it will give red color, in the ionized state it will give blue color. So I hope this is clear to you, and it will remain in the unionized state in the presence of hydrogen ions, and it will remain in ionized state in the presence of hydroxide ions. So uh, this was our first theory. Now let us begin with the next part, which is the Oswald's theory. Okay, so this theory states that the color change of the acid base indicator is the result of its ionization, because the ionized and the unionized forms are different in color. Okay, so this is also quite similar to the litmus one only. Okay, so as the acid base indicator is either a weak acid or a weak base, its ionization is highly affected in the presence of acids and bases. So let us understand this with an example. So for example, if we say that the indicator is a weak base, if already we are having a indicator which is a weak base, then its ionization in acids is very slow, because there are common H positive ion, and its ionization will be fairly well in the alkalis. Similarly, if the indicator is a weak base, then its ionization in acids will be high and in bases will be low, because whenever there are common ions in our solution, the ionization always reduces. Okay, so this is what is happening over here. Let us understand this with an example. This theory will be clear to you with this example. So let us talk about phenolphthalein. So in a solution, it undergoes ionization to a small extent only. Okay, so here I have given an equation. So this is our phenolphthalein. So whenever it is in unionized state, it is colorless, and when it will be converted into ions, okay, this is the ionized state, this is the unionized state. So in unionized state it is colorless, in ionized state it is pink. Okay, so what is happening, that the colorless phenolphthalein molecules are undissociated and pink colored. Okay, and pink colored are the pH negative ions. Okay, so what is happening, that in acidic solution the ionization of phenolphthalein is negligible, as the high concentration of H positive ions shift the equilibrium towards left. Okay, so whenever the phenolphthalein is put in acidic solution, the acidic solution is already having high concentration of H positive ion, therefore the equilibrium is moved towards this direction, and the phenolphthalein stays in the undissociated or the unionized form, therefore the solution is colorless, which means that in acidic medium the color of phenolphthalein is colorless. I hope this is clear to you, because it remains unionized. Now what happens in the alkaline solution? Now in alkaline solution, the OH negative ion remove the H positive ion in the form of water molecules, which shift the equilibrium towards the right. As a result, the concentration of pH negative ion increases in the solution, imparting pink color to it. pH negative ion is the ionized form of phenolphthalein. So what is happening, that when phenolphthalein is put in the when phenolphthalein is put in alkaline medium, so this H positive ion are being taken away by the OH negative ion. Okay, so phenolphthalein from the unionized form changes into ionized form, and in ionized form it is having pink color, in unionized form it is having it remains colorless. Okay, so this is you will, I think you understand this with this example, that how in acidic and in basic medium the color is being imparted. It is due to the ionization. Now the next theory, it is the quinonoid theory. So this theory is quite different. So it says that the color change of indicator is due to the structural changes of indicator. Now there are certain changes taking place in the structure of the indicator that is imparting the color. So in acidic solution, indicator exists in different structural form, and in basic solution it exists in different structural form, and these structural forms have different colors. Therefore in acidic medium indicator give different color, and in basic medium it give different color. Okay, so what exactly happening is this, that it says that the indicators are having different structural forms in acidic and basic medium, and due to the different structural forms the different colors are being imparted in the respective mediums. So okay, so this is the benzonoid form which is having different color. This is the quinonoid form which is having different color. Now let us see an example and you will understand this theory better. So we will again take the example of phenolphthalein. So what is happening, that phenolphthalein is in benzonoid form in acidic solution and is colorless, but in alkaline solution it is converted to quinonoid form which gives pink color. Okay, so here also written the equation for this. Okay, this is the benzonoid form of phenolphthalein. The benzonoid form of phenolphthalein is okay, in acidic medium the phenolphthalein is present in benzonoid form, and benzonoid form is colorless, whereas when it is being transferred to alkaline medium it is converted to this quinonoid form, and the quinonoid form is pink in color. Okay, so due to changes in the structure, different color is being imparted in different mediums. So this is what we get to know from our quinonoid theory. Okay, so in this video we covered the theories of acid base indicators, which are different from the theories of acid base titrations. Okay, this is a very important topic. Often questions are being asked from here. So I hope you liked the video, and if you did, please subscribe, and thanks for watching.